Hello students, welcome to chapter 14. Today we're going to be talking about DNA structure and replication. This is basically talking about the discovery of how scientists discovered the structure of DNA, how they discovered that DNA was the genetic code of living creatures, and how they discovered that replication was happening inside DNA. So first of all, DNA is deoxyribonucleic acids, right? It is a phosphate plus a nucleic acid plus a base sugar molecule. Rosalind Franklin did the discovery of how it's kind of designed structurally, and then Watson and Crick had the chemical structure of how it's designed. So the DNA molecule revolution was launched within biology after that point, making it possible to relate genetic traits of living organisms, discover the molecular code of DNA in every cell. And then much later after that point, they developed PCR to try and develop molecular genetics, to try and relate that to taxonomy, phylogenetics, everything like that. Okay. So Rosalind Frank and to a lesser extent Maurice Wilkins of King's College London used an X-ray diffraction. So this is a crystalline X-ray machine to study the DNA structure. Franklin interpreted an X-shaped distribution of spots in the diffraction pattern to mean that the X-ray has an helical structure. So it has this helical bend to it to try and warp all the DNA into a more compact molecule. So X-ray diffraction, this is a beam directed at the molecule to form a solid, ideal crystalline form. Basically, it's like shadow puppetry. So you form a solid beam towards a very, very small sample, and then it basically forms a shadow towards a photographic plate. Positions of atoms in the molecule are deduced from diffraction patterns produced on the photographic film. So anything that's not, covered up by that DNA sample is shown as white, and then the DNA sample is shown as a shadow. Watson and Crick, after that, constructed a double helix model for all the chemicals inside that structure. So people beforehand found all the chemicals inside that structure and found out from Franklin's X-ray data that the polynucleotides inside of it needed to be in more of a helical kind of spiral staircase structure. The two chains are anti-parallel. So one chain goes in a 5 to 3' direction, and the other chain goes in a 3' to 5' direction. And that's all dependent on how the carbon is formatted inside the sugar molecules. The deoxyribose molecules. The pairs of bases fill the central space. So inside those central spaces, what you call, if it's a spiral staircase, is the stairs. Those are the bases. So the adenine, thymine, cytosine, and guanine, those are the bases. So all of that satisfies the Franklin X-ray and what's called the Chargaff's chemical analysis. So the Chargaff rule is something that we'll explain in just a minute. Many scientists once believed that proteins were the hereditary molecules. So before all this, before that we found out that DNA was the hereditary molecules, they thought that proteins were. In 1928, Frederick Griffith found out that a different type of substance, DNA, or nucleic acids, could genetically transform bacteria. And this is called the transformation principle. So he studied two different strains of Streptococcus pneumoniae. Okay, one's called a smooth strain, which is highly infective, otherwise called virulent, and then a rough strain that is non-virulent and does not cause a lethal dose. So the smooth strain is called S, and the rough strain is called R. Some of these bacteria can interact with each other, so the smooth strain can cause the rough strain to become virulent. Some molecules released when the S cells were killed could change living non-virulent cells genetically to the virulent S form. So sometimes if you have an S cell with an R cell, it can change that R cell to an S form. And they called that the transforming principle. Right? So it can change that R cell, if you have an S cell, to another S cell, and that's changing that to an S cell through DNA. Right, and that's through its pili, and it just basically exchanges DNA to the R cell to make an S cell. So if you see on the image here, if you inject a mouse with a rough strain and an R cell, it lives. It's fine. If you inject it with a smooth strain, it dies. Right? That's your controls. If you inject it with a heat-killed smooth strain, it's heat-killed. It's no longer working. Right? The mouse lives. However, if you inject it with a rough strain and heat-killed smooth strain, keep in mind, it's heat-killed. The proteins are no longer working in that heat-killed smooth strain. However, the DNA is still there. So the DNA is still there. The proteins are no longer working. The DNA is still there. The DNA can still transfer to the rough strain. So it still can be transferred to the rough strain, making it lethal. So the mouse dies. That's called the transforming principle. So the smooth strain is no longer working. It's no longer active in, so to speak, it can no longer produce proteins. It can no longer produce anything to make proteins, but the DNA is still there. So the rough strain can produce proteins. So it picks up the DNA from the smooth strain. So it's producing proteins now from the smooth strain, and it's now producing the lethal dose. That's called the transforming principle. That's what it does. It transforms all of the DNA from the other cells. And this is something that bacteria does. So we ourselves can't just ingest something and produce its own DNA. There are other things that can do that. There's a type of snail that can do that, but we ourselves have lost that ability. Anyway, there's another experiment that goes kind of further into this. In 1940, Oswald Avery identified the chemical nature of Griffith's transforming principle. So Avery broke down the heat-killed S bacteria, the virulent bacteria, and destroyed the class of molecules. So he broke them down into protein, the DNA, or the RNA. And he wanted to make sure that he knew which type of molecule was being transformed here. When the proteins or RNA was destroyed, the extract still transformed our bacteria into virulent S bacteria. But when DNA was destroyed, no transformation was occurred. The transforming principle was DNA. So you have some heat-killed virulent bacteria. You have some S strain that's heat-killed. And if you put it into protein, if you put it into lipase, you put it into carbon or carbohydrates. And then if you put it into an R strain, what's going to be left is you're still going to have the S and R strains, right? Because nothing has changed. All of these still contain the same amount of DNA, the same amount of proteins, the same amount of anything that's still within it. However, if you put DNase in there, if you put an S strain, if you put DNase in there, and then if you put the R strain, if you put it into the R strain, the S strain has been killed by the DNase. Why? Because you've now killed the ability for it to transform to the R strain. You now killed the ability for the S strain to transform the DNA over to the R strain. To transform that DNA to the R strain. So now all that's left is the R strain. So the S strain is now killed. The S strain is now gone. Right? And all that's left is the R strain. So no transformation occurred when the DNA was destroyed. So you don't have any S strains anymore. You don't have any virulent strains anymore. The next one that they wanted to do is the Hershey and Chase experiments. They wanted to really make sure that the DNA was the transforming principle. So in 1952, Alfred Hershey and Martha Chase wanted to see that DNA was the hereditary principle, the hereditary molecule. Hershey and Chase studied a virus, basically a bacteriophage or a phage. This is a bacteriophage. Bacteriophage or phages in general are viruses that attack other cells. So a bacteriophage is a virus that attacks a bacteria. So this one affected E. coli. The T2 phage, studied by Hershey and Chase, consisted of only a core, so a core consisting of DNA, surrounded by proteins. So you have the core of DNA and proteins surrounding that. So the phages were either labeled with proteins, with an S35, that's an isotopes, or their DNA with P32, that's another isotope. So the protein on the outside had S35, or the DNA inside had P32. That way they could tell the difference. Either the protein on the outside was labeled or the DNA on the inside was labeled. So after the tests, they could show that either the protein got into the cell or the DNA got into the cell. So over on the side, you could see the proteins were sloughed off and the DNA was inserted in. So they showed that the labeled DNA, not the labeled protein, entered the cell and appeared in progeny phages. So the DNA got into the cell, and then later the DNA was split, multiplied over and over again, and then later they saw that the DNA was in later offspring of the viruses. So the experiments of Griffith, Avery, and his co-workers, and then Hershey and Chase, established that DNA, not proteins, carries genetic information. This research also established the term transformation, or the transformation principle, the alteration of a cell's hereditary type by the uptake of DNA released by breakdown of another cell. So basically, transformation, putting out your DNA, releasing that, breaking up another cell, and then building that DNA, splitting it apart, producing offspring in that manner. Okay, now we're going to talk about the structure of the DNA. DNA contains 4 nucleotides, all consisting of 1 5-carbon sugar, deoxyribose, 1 phosphate group, and then 1 of 4 nitrogenous bases. DNA is not a sugar. DNA has sugar in it, but DNA is a nucleotide. DNA is it's a nucleotide. It is a specific type of carbohydrate, right? It's not a sugar, it's a carbohydrate. 1 of 4 nitrogenous bases, we have adenine, we have guanine. These are both purines, right? They have 2 fused carbon nitrogenous rings. Nitrogenous rings because they contain nitrogen. And then we have thymine and cytosine. Both of those are pyrimidines. Pyrimidines, they have a single carbon ring. One of the ways that I can think to kind of memorize this, all gold is pure. So adenine, guanine, are purines. So all gold is pure. And then the other ones are pyrimidines. So Chargaff's rule. Chargaff's rule, to begin with, A goes to T and G goes to C. And this was basically thought up because when you take a amount of DNA, all adenine equals up to thymine, and then all cytosine equals up to guanine. But guanine always binds to cytosine, and adenine always binds up to thymine. Hypothetically. That's the way it's supposed to go. Okay, that's Chargaff's rule. All right, DNA nucleotides are joined to form a nucleotide chain, polynucleotide chain. Right, so DNA is double-stranded. We have the deoxyribose sugars. All of those are linked to a phosphate group in an alternating pattern. We have phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. That's called a sugar-phosphate backbone. All of those go from 5' to 3'. So it goes 5 to 3, 5 to 3, 5 to 3, 5 to 3. At one end, the phosphate group is bound to the 5' carbon of the deoxyribose sugar. Okay, that's your 5-carbon sugar. 1, 2, 3, 4, 5. And you can see it on the list there, on the red markings on the picture. 1, 2, 3, 4, 5. That's the 5' carbon. At the under end, the hydroxyl group is bound to the 3' end of the deoxyribose sugar. So that's the 3' carbon of the phosphate group, the hydroxyl end. So a phosphate group is bound to the 5'. The other end, the hydroxyl group, is bound to the 3'. The entire linkage is called a phosphodiester bond. It's called a phosphodiester bond because you have two esters there. Right? You have 1 O and 2 O. Two oxygens binding that phosphate group. It's always going to bind 5' to 3'. So if you look at the other end, the other side, you can see it actually going backwards. 5' to 3'. That's why it's called anti-parallel. So 5' to 3' going from up to down, and then 5' to 3' going from down to up. This is important because when we talk about replication, it's going to be going the opposite direction. Antiparallel strands at 3' hydroxyl group is exposed at the newest end of a new DNA strand. The oldest end has an exposed 5' triphosphate. Right? So we have the DNA polymerases. They only assemble at 5' to 3' direction. So you're going to have this 5' to 3' going real fast. But because DNA strands run anti-parallel to each other, the template strand is reading into 3' to 5' direction. Okay? So that one is going 3' to 5' direction. So how do we fix this? I would tell you when we get to the DNA replication part of this. Really quick, let's talk about Mendelssohn and Stahl experiment. So how did they figure out how replication is being enacted? In 1958, Matthew Mendelssohn and Franklin Stahl of California Institute of Technology demonstrated that DNA replication is called semi-conservative. Basically, they used a non-radioactive heavy nitrogen isotope, it's called N15, to tag parental DNA strands. So this is another marker that they use. So just like in the other experiment, they used a marker to try and figure out where these molecules are at. So they used a nitrogen isotope, N15, basically to say, where are these strands being put at when they're being replicated? DNA molecules with different densities were separated by centrifugation. So when it's too heavy, when it's heavier than the other DNA, it's going to separate when it's centrifugated. Right? So it's when it's spun, the heavier ones are going to go down faster than the lighter ones. So let's say that we have some type of conservative replication. If you have a conservative replication, you have two strands of original molecules serving as templates. Right? So the two new strands of the DNA molecule will unwind, and then two old strands of the molecule will be put into place. So two new strands will be put into place, and two old strands will be placed. That's called conservative replication. Basically, all of the old DNA is conserved, and then you have two new strands completely made. And then the next generation, you have all of the old DNA and all of the new DNA, and then the new DNA, you have all of the new DNA and then all of the new DNA again. What's the problem with that? You have one cell that has all the new DNA, all the old DNA just sitting there. You're just gonna have all old DNA. Well, what happens to the old DNA? It may degrade over time. It may cause problems to that old cell. So the old cell may just die. We have dispersive replication model. Neither parental cell is conserved. Both chains, either replicated molecule, contain old and new segments. So this one is a little bit weird. Basically, you have this splitting of the molecule of DNA. Just some new, some old parts of the DNA happen to be in different places in each of the new DNA molecules. And then in the new generation, you have, again, different parts of the new and old DNA molecules. Well, what's the problem with this one? You almost have no conservation of any of the DNA. You could have mutations going left and right and not even know what could be a problem. So in this one, you could have a lot of error. You could have a lot of error and not even know what could be a problem. In this one, it's called a semi-conservative replication model. This one is based on complementary base pairing. and explains DNA replication. Hydrogen bonds between the two strands break, the two strands unwind, and they separate. Basically, you have two different strands, they unwind. Each strand acts as a template for the synthesis of a new complementary strand. So one strand acts as a template for a new strand, the other strand acts as a template for the other strand. Each new double helix has one old strand derived from the parental DNA molecule, and then the other strand is a new. So this just kind of makes sense. One strand is old, one strand is new. That's why it's called semi-conservative. One strand is conservative, the other strand is new. So now we're going to talk about what happens during DNA replication. So RNA polymerases can add new nucleotides only to existing strands, right? So this explains why there's an old strand available. DNA polymerases must add to an existing strand. There must be an existing strand available to be added. Because two new strands of a DNA molecule are anti-parallel, only one template strand runs in a direction that allows DNA polymerase to make a continuous 5' to 3' copy, right? And this is called a leading strand. A new strand begins only with a short chain of RNA synthesized into an enzyme. Basically, a primer has to be primed by a protein. And this is called primase. Okay, so you have primase priming where it starts replication. DNA polymerase copies the other strand in short lengths. This is the lagging strand. So it copies it in short lengths. This is called Okazaki fragments. And it's synthesized in the opposite direction to that DNA unwinding. This is called discontinuous replication. So these little fragments are copied just in short lengths, in little fragments, and that's called discontinuous replication. We have DNA polymerases. They assemble complementary polynucleotide chains from individual deoxyribonucleotides. 4 different deoxyribonucleoside triphosphates, that's hard to say, basically DTPs, one for each DNA base. Basically, you have DATP, DGTP, GCTP, and DTTP. So DNA polymerase adds all of these at a 5' to 3' direction. Right, so you have all of these added up, and if it's the leading strand, it just goes like a zipper, it goes vroom. If it's the lagging strand, it goes doop, 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 doop. So all of these have to be added to an existing nucleotide chain. DNA polymerase structure. It's consisting of different types of subunits, all arranged in kind of a hand-shaped domain. Template DNA lies over the palm in a groove formed by the fingers and the thumb. And remember, whenever you add anything to another molecule, you have to use energy. all of those triphosphates has to be using energy, right? So it's polymerization. Okay. The template strand and the 3' hydroxide of the new strand meet at the active site, where that protein is, at the polymerization reaction of DNA synthesis located in the palm domain. And this is happening like really fast. So just going. There's other proteins involved. So one is a sliding DNA clamp. As DNA polymerase extends the new DNA strand, one nucleotide at a time, it moves it along the template. The sliding DNA clamp encircles the DNA and attaches to the rear of the DNA polymerase. And it has to tether it to the template strand, right, to make sure that it stays on. It also increases the rate of synthesis. Basically, it increases it 1,000-fold compared with a non-processive polymerase. Other ones, you have to unwind the DNA. So helicase unwinds the DNA into a Y-shaped replication fork. So it comes through and unwinds the DNA. Helicase forms what's called a replication bubble. And then single-stranded binding proteins keep it from pairing back up again. Right? So it keeps it from pairing back up again. Topoisomerase. These can cut and rejoin the DNA, preventing twisting in circular bacterial chromosomes. Topoisomerase can also happen in eukaryotes, but these usually happen in prokaryotes. So remember I was talking about leading strand and lagging strands. The leading strand is synthesized in the direction of the DNA polymerase, of the DNA unwinding. The lagging strand happens to the 3', 5' direction. And this is a discontinuous strand, right? So this happens with the Okazaki fragments. The DNA helicase unwinds all the DNA. The primase initiates all new strands. We have different types of polymerase. Polymerase 3 is the main polymerase. That's the one that initiates the leading strand. Polymerase 1 forms the lagging strand. And then you have ligase that binds the Okazaki fragments together. So here's some pictures that go along with that. You can see the replication fork. We have the leading strand. You have primase at the top that's putting on those RNA primers. DNA helicase is unwinding that DNA, making that bubble. And then you have single-stranded binding proteins that's helping that separate apart, keep it separated. And then we have the leading strand that the DNA polymerase is leading all of those DNA to bind on to the DNA of the old model. DNA polymerase 3 binds the leading strand and the lagging strand, but the leading strand continues and the lagging strand is in this continuous model. And you can see that down here, where the DNA polymerase 1 in the leading strand is very continuous while the lagging strand has nicks in it. So you have first Okazaki fragment, you have second Okazaki fragment. And on the bottom, we have a nick. And that yellow is DNA polymerase 1, right? And it'll help to get rid of the RNA primers, right? So DNA polymerase 1 will help to get rid of those RNA primers and then polymerase DNA onto that segment. And DNA ligase will actually bind together the DNA polymerase, those Okazaki fragments, together. Now we have some videos to watch, some YouTube videos. I would suggest you go through and see if you can find the attendance assignment here, okay? If you watch one of these, or if you just look online, there are 3 different phases of DNA replication. All right, 3 different phases. What are those 3 phases of DNA replication called? This is your attendance assignment. What are those 3 different phases of DNA replication called? We're going to talk about telomeres. There's an end to your DNA called a telomere, and this is just a repeating section of the 3' end. Okay, this is just kind of a repeating end where it's just over and over again, this repeating end that's it's non-protein based. It doesn't make anything, it doesn't make a protein or anything. It's a buffer of non-coding DNA consisting of short repeating sequences. So telomerase stops the shortening of telomeres, right? Basically, as you age, every time your DNA replicates, it kind of shortens that repeating sequence. And as it shortens, it gets closer and closer to a coding sequence. Okay, and that's bad. As it gets closer to a coding sequence, it can mess up proteins that might cause issues down the line. Telomerase stops that shortening. It can add telomere repeats to the chromosome ends. Most of the time, this is active only in rapidly dividing embryonic cells or in germ cells. So if you're an adult, you don't really have those embryonic cells. If you're an adult, though, these can happen in cancerous somatic cells. So, yeah, it's not really a good thing. There are scientists who are trying to figure out the idea of telomerase and trying to lengthen the aging process or shorten the aging process, but it's a long way off from now. Okay, so during DNA replication, there's called a proofreading system that allows polymerases to back up and remove mispaired nucleotides. So we do know that A goes to T and C goes to G. Sometimes the molecules can get switched up. If a newly added nucleotide is mismatched, DNA polymerase can go back and proofread it. It can go back and reverse using a 3' to 5' exonuclease activity to remove the newly added incorrect nucleotide. So if A suddenly turns and pairs to C for some reason, it can go back and remove that A and pair it to G. It then resumes forward synthesis and inserts the correct nucleotide. So it does that just in case there's some type of mutation problem or something like that. It doesn't always work. Sometimes proofreading doesn't always work and there is a mutation. But proofreading does help in case, mutations, it doesn't want there to be a mutation. These are the types of repairs that can stop mutations. DNA repair mechanisms correct base pair mismatches. So distortions in DNA structure caused by mispaired mutations. So these are A to C or T to G or whatever. Base pair excision. Basically, you have a non-bulky damage by removing erroneous base and replacing it with the correct one. So if you have a mispair where the nucleotide is removed and there's basically an excision and there's basically no nucleotide there, it can go through and replace it with a nucleotide. Nucleotide excision repair. This one is actually where there's a bulky distortion. So, such as thymine dimers. So, errors that actually make a adduct. So it makes a bulky adduct in it. So it actually has to unwind and then bring it back in. This is actually another chemical imbalance. And then there's errors that remain in DNA after proofreading. So this is one that can actually cause a mutation. Mutations in a gene can alter a protein encoded by the genes, which can alter how the organism functions. So this is what I was saying. These types of repairs don't always fix in proofreading. So there's single-stranded, there's also double-stranded. End rejoining, sometimes it just splits the DNA. Sometimes it actually has to pair it back together. And then there's template-based repairs. If there's some type of splitting and the template actually splits in two and there's a kink in it, it actually has to go through, repackage it, and then split it apart, bring it back together. It's actually really weird. Make sure to go through and do the attendance assignment, and then I will get to everything else very soon. All right, let me know if you have any questions. I will see you later.